So quickly, this is the agenda we've proposed for today for the next just under an hour. You guys are introducing yourself. We'll introduce ourselves. We'll do a quick overview of the platform and where to locate both the lessons and the new resources that are on there. Donna was mentioning those biomimicry organism cards, which we have a complete set now, so we'll show you where to find those. And then we've selected 2 lessons to do a deep dive into the physics lesson, which is Built for Brilliance on structural color, and the chemistry lesson on anhydrobiosis called the African midge and vaccine stabilization. And then we'll just do a quick review of Global Design Challenge, where you can find the resources, and we'll take questions. Great, thank you all so much for introducing yourselves. Thanks, Lisa. So we seem to have a mix, but hopefully wherever you're at, whether you've introduced or you've taught a lesson or you haven't started yet, hopefully you can get something out of this as you move forward. So quickly, I'll introduce myself. My name is Abby Randall. I'm the program manager for EcoRise, and I also helped write some of this curriculum. And it's one of the most robust, beautiful curriculum I've ever seen, so I'm really excited to share more of it with you today. Gretchen, would you like to introduce yourself?
Yes, sorry, it took me a moment to get myself off mute. Hi, everyone. My name is Gretchen Hooker. I am the Education Resources Manager at the Biomimicry Institute. My background is in sustainability and industrial design specifically, and I worked with Abby and her colleagues on this curriculum, serving primarily as a biomimicry subject matter expert for the science lessons and also as the lead author for the design challenge module. And super excited. It's one of the most full-featured curriculums that we've produced at the Biomimicry Institute, and really excited to be doing that with EcoRise.
Awesome, thanks, Gretchen. So what we're going to do is I'll sort of be your logistical and instructional person, and Gretchen will be our kind of science biomimicry person, and we'll alternate in the way that we present information. And we're a small group, so please jump in at any point if you have questions. Don't hesitate. So I just want to orient everybody on my screen. This is the home screen that you see when you log into our biomimicry and science platform. And so you've all done that orientation, I know. We have a few new resources. I just want to make sure you are aware of them. So under the Introduction to Biomimicry, this actually just went up about 2 hours ago. The Biomimicry Institute has created this guide for K-12 educators for kind of sharing and teaching biomimicry. So I'll just show that off to you here for a second. Feel free to follow along if you happen to be in Canvas. But there are these guiding principles from biomimicry that are threaded through all of the lessons and are the foundation. And we talked about them a lot as a curriculum writing team, but they weren't wholly apparent, and so we wanted to create something that really outlines those threads. And so here we're looking at structure, function, and strategy. We're looking at patterns, scale, systems thinking. So this is a document you can read whenever you're looking for a little bit of inspiration or some foundational knowledge in terms of teaching these lessons. And then down at the bottom, you'll see we have our chemistry lessons, we have our physics lessons, and our biology lessons. And then below the Biomimicry Design Challenge, which we'll come back to, are inspirational resources. And so these are those inspired by nature cards that Dorna was mentioning. They're a complete set, they're updated, so you can download those. We've also produced this set of inspirational biomimicry posters. So they're there for you to download. There are 5 of them. So just to show you a quick example, this is just one example of a Jeannie Benia's quote. If you have a color printer, you can print these and put them up around your classroom, or email them out to your students, or however you'd like to share them and provide some biomimicry inspiration. Okay, and then there's a few other resources down here, just in case you haven't seen them. TED Talks, handbooks, videos, links, just things to give you a little bit more background information if that's what you're looking for. So the first lesson we wanted to dive into today with you is a physics lesson. And it is this Built for Brilliance lesson on structural color. And so we'll just orient you a little bit to the lesson, and then Gretchen's going to share a little bit about the science behind the lesson. So on every page, there's a nice summary here, and then you can download the lesson plan, the presentations. This is an example of a lesson plan. This is the Built for Brilliance lesson plan. So essentially, this is broken into two lessons. So the first lesson is just your classic double slit experiment. So you're looking at what is light, and you're examining it, and you're conducting that. It's a very classic experiment that people are doing in their physics classroom anyway. What's really nice is we're using it through the lens of biomimicry. So you do this lab with them. There's a great video to show them the classic experiment when you're finished. And then in session 2, you dive into this beautiful presentation, and I'll show it off here for you just for a minute, just to show you how beautiful these images are. So we have this entire slideshow, which really, covers a lot of those physics standards that kids, that we have to teach in a physics classroom, and then incorporates and applies biomimicry. So looking at wave interference and, what is color and the electromagnetic spectrum. And you'll notice that there are detailed notes to go with each slide if you need some of that background information in terms of what is color. So I wanted to just point out to you on this lesson plan, as you go through, it gets more and more specific. And so after you're looking at what is color and exploring that, then the lesson asks students to have a little design challenge and look at how this is applied in industry. So Gretchen, I'll turn it over to you here to look at some of those biomimicry examples of structural color.
Sure. So that lesson ends actually with a really interesting video produced by Veritasium that talks about one of the applications of structural color. And, hang on a second, I'm going to share my screen. It's a little hard to talk while I do that. Let's see here. Okay, I think you should be looking at my web browser. So the biomimicry innovation that's featured in this lesson is a anti-counterfeiting and security technology created by a company called Nanotech Security. And there's a video, excuse me, linked in the lesson that does a really great job of explaining how that technology works. And I just wanted to take a moment to show you where you can find some additional information on Ask Nature about that technology, as well as some other technologies that are inspired by the same phenomena in nature, the morpho butterfly structural color. So the nanotech security technology, like I said, is an anti-counterfeiting technology, and it works by taking a thin film of material and punching a lot of tiny little holes in it, which serves that purpose of creating constructive and deconstructive interference such that it reflects selected colors, and it creates this sort of iridescent reflective image on the surface that it's these holes are put into. And so it can be used to make banknotes very hard to counterfeit. It can be used on bank cards and other sort of essential security things, or place like tickets that you don't want to be counterfeited. So just by way of using this example to show you a little bit around Ask Nature. So this is a inspired ideas page on Ask Nature. Ask Nature, from the landing page, you'll see has 4 main content types, and those are biological strategies, inspired ideas, resources, and collections. And these different content types have relationships to each other and are connected in a variety of different ways. So, for example, from this inspired ideas page on the anti-counterfeiting technology. We can see over here in the right-hand side how it is linked to a biological strategy about the structural color of the morpho butterfly's wing. So if we click on this, it should open the morpho butterfly biological strategy page. And you can see here the headline is "Wing Scales Cause Light to Diffract and Interfere." And the organism in question, morpho butterflies, shows up beneath this statement, which is essentially getting at what the strategy is doing. There's a little bit of a summary headline here that explains that just a little bit more, a variety of engaging images that show the organism, diagrams that help explain the phenomena that's occurring, and occasionally micrographs and other images from research that also provide some really interesting scientific images. Our strategies also typically all have a non-technical summary that describes what's going on with that strategy in nature. Here, you could, there's an embedded video. This is actually the same video that appears in the lesson. And then all of our strategies have links to scientific references. So if you were interested in learning more, or were working with advanced students that want to actually get into scientific journals that are talking about the phenomena, we have selected references also as part of these biology strategy pages. So again, our biological strategy pages, if they have been, if that biological strategy has been applied to the creation of a bio-inspired innovation, you will see those inspired idea pages, if they've been created yet on Ask Nature, and we are still building out the content, but if they have a record on Ask Nature, it will show up here in the right-hand side under Inspired Ideas. And you can see that the morpho butterfly has inspired a lot of different innovations. So here at the top of the list is the nanotech anti-counterfeiting security technology, and a variety of others. And I was going to take some time to just talk about a couple of those with you today. So this one here is a product called Morphotax, created by Tasian Fibers Limited, and it is a fabric that is colored without any chemical dyes. So it's actually a colorless thread, but because of the structure, the variations in thickness and texture of that colorless material, it actually creates this iridescent, shimmery blue color, much like the morpho butterfly. And you can see here a garment made out of that, that textile. And going back again here, another really interesting technology is Mirasol, which is an electronic display technology. Now, this works very differently than other display technologies, because instead of producing light the way a backlit LCD display works or an LED panel display, Mirasol screens reflect ambient light in the environment. And so they're able to operate with very low power because they're not running a light source. And they have the added advantage of being viewable in bright sunlight conditions. I think we probably all had the experience of being out in the bright sun and sort of struggling to see what's on our phone screen. The Mirasol display technology doesn't have that disadvantage, because that bright sunlight actually makes it more visible instead of less visible, because it's capturing that and reflecting back that ambient light. And so the way this works is it's made up of tiny little units that are composed of two plates with an air gap in between them. And we have a video that I wanted to show you that explains how that works. I think Abby's actually going to play it from her computer, because it should be a little more stable than trying to stream it from the internet on my computer to you. So I'll let her cue that up. The way this works, the light is passing through the top plate and reflected by the bottom plate, which produces constructive interference and produces color the same way that those nanostructures on the surface of the butterfly wing work. And only a very small electrical charge is required to open or close that gap, and so it's able to operate with very low power and only requires energy to change the state of having that subpixel on or off. And this explanation will make a lot more sense once we can pull up the video. There we go. I think it should be showing now. There'll be a voiceover here in a moment. Abby, I'm not hearing it.
The interferometric modulator element, also known as an IMOD element, is a simple micro-electromechanical system device, or MEMS device, that is composed of two conductive plates. One is a thin film stack on a glass substrate. The other is a reflective membrane suspended over the substrate. There is an air gap between the two plates. When no voltage is applied, the plates are separated, and light interacts with the open elements to reflect a specific color. When a small voltage is applied, the plates are pulled together by electrostatic attraction, and the light interacts with the closed element to appear black. The IMOD element has two stable states, open and closed. IMOD elements are minuscule, typically 10 to 100 microns on a side. The color of the IMOD element is determined by the size of the gap between the plates. As shown, the red, green, and blue IMOD elements have air gaps of differing sizes. By manipulating each minuscule pixel, a wide spectrum of color generation is possible. Each subpixel moves to either an open or closed state to create the overall pixel color. To create a flat panel display, a large array of IMOD elements are fabricated in the desired format and packaged. Strips of IMOD elements are combined to form pixels, and pixels are combined to make the overall display.
Gretchen, did you have anything to add?
Yes, I'm sorry. I was speaking while I was muted. Oops. I was just saying that, I think this is a really, really interesting and exciting application of this technology because it's so relevant to our daily lives in that most of us interact with screens on a daily basis that require a lot of electrical energy. And so it's pretty interesting to think about how that could be completely different by applying this strategy that comes from, this beautiful morpho butterfly phenomena in nature. Just also to add to your sort of toolkit of things to learning and thinking about structural color, further down this strategy page here, you'll see there's a link to an article on Zygrowth Quarterly, which is a really wonderful quarterly journal on biomimicry. And there are frequently really wonderful in-depth case studies of different technologies in Zygrowth Quarterly. And in issue number 5, they did a wonderful overview of Mirasol and actually some struggles that the Qualcomm company encountered in taking this technology to market. It is not yet widely available in the marketplace, but there are some hopeful things on the horizon. I did read up in advance of this webinar. There are some rumors that Apple may be in the process of acquiring this technology or trying to take it a little bit further down the line towards being more widely adoptable into conventional consumer electronics.
Gretchen, I think you had to re-enable the page. Did you want to show them something right then?
Yeah, I'm so sorry. Here we go. Show this window. Yeah, so we just watched that video, and I was just pointing out that below that there's another section of this innovation page, Biomimicry Story, and you can see here this is where the link to the Zygote Quarterly article can be found.
And I think there's a really nice environmental lens here that you can use with this lesson, talking about, why might we want to use structural color instead of, say, paint, and talk to kids about the toxicity, of paint and the benefits of using structural color in its place. And that can lead to interesting conversations. Does anyone, before we switch gears into our midge lesson, does anyone have any questions while you have Gretchen and myself here to answer any questions about lessons or the science behind this concept? All right, I'll take that as a no, but if something comes up, feel free to chat it in there, or we'll take questions again at the end. Gretchen, was there anything else you wanted to add before we moved on?
No, I think we're ready to go.
Okay. All right, let's put it back on my screen. All right, so that was a physics lesson, and now we wanted to jump into a chemistry lesson. So I'll just remind you where to find it. The first module is actually chemistry, and the fifth lesson is the one that we wanted to dive into. This actually wasn't available for anyone who attended our last webinar. This was one of the last lessons to be complete, so it's now fully available to you. And so just like all of the other lessons, has a summary here. So this is looking at how the African midge in northern Nigeria uses anhydrobiosis in drought conditions to survive, and how to apply this to vaccine stabilization in disaster zones. So just like the other lesson plan I showed you, this lesson is divided into multiple parts. And so it actually starts with an article that looks at the midge, and then it leads into a solubility lab, which aligns really nicely with chemistry standards and allows students to examine stability and solubility. And so those include, here you can see you can download the lab, and there's also sample answers available so students can check their work or compare answers as they're going through the lab. And so what I really like about this lesson, so after going and explaining through how the midge survives and what is cryptobiosis and what is anhydrobiosis, the students are asked to imagine that they are in a small, underdeveloped country that's been hit by a tsunami, that hundreds of people have died, and many more are vulnerable to diseases such as tetanus. And it challenges them to think about how they could apply what they've learned with the midge and anhydrobiosis to develop vaccine technology so that these vaccines can be stable and last, in extreme conditions with heat. And so they can be delivered in those disaster zones. And so it's a great, it looks at the existing technologies and gives them a small challenge for them to, kind of these small challenges in each of the lessons prepares them for the larger global biomimicry design challenge that many of you will choose to have your students compete in. Let's see. Gretchen, I'm going to turn the screen over to you to navigate through that Ask Nature.
Okay. Okay, so providing again a little bit of context for the lesson and what additional resources are available on Ask Nature. So sort of the context of the challenge presented to students in terms of why anhydrobiosis might be useful in a innovation that solves a problem. there is an existing technology created by Nova Labs that applies the anhydrobiosis concept to the preservation of pharmaceutical products in a non-liquid form. And they have two related technologies that do this, the vitreous and the hydrous vaccine stabilization technologies. And the way these work, the hydrous is preserved in a dry format on a sort of non-woven textile type product, which you can sort of see illustrated here in this still from a video on the Ask Nature Inspired Ideas page. And it's actually hydrated upon delivery. So the active agent in the vaccine is encapsulated in a sugar molecule, much the way that the African midge preserves its tissues when it dehydrates. And then using a sterile water solution in a syringe, that stable vaccine in the sugar molecules in a dry format is rehydrated as it passes through that sort of wafer-looking attachment on the end of the syringe before it gets to the needle. And so it's delivered in a hydrated form to the patient. They also have the vitreous technology, which is the same type of sugar stabilization, except it's suspended in a non-hydrous, so not water, liquid, which is then injected, and then the body is able to dissolve the sugar in the vehicle for the vaccine and make, once that's dissolved, then the vaccine itself, the active agent, is readily available in the body. And so I believe they use, a sterile oil type substance in that case. And so you can see here on Ask Nature, this vaccine stabilization strategy is linked to a couple different biological strategies on Ask Nature that exhibit the anhydrobiosis phenomena. And one that I want to talk with you about today is the tardigrade, which also has the cute nickname water bear and sometimes moss piglet. And they're called that because they kind of have this cute rotund form with apparent claws, and they live on mosses and lichens. And they are able to survive really extreme environmental conditions by entering that state of anhydrobiosis, where metabolism essentially ceases and the organism shrivels up in a desiccated form. And as mentioned earlier, the African midge uses a sugar to preserve its tissues, and the water bear is using the same or a very similar sugar to do that. And so when it encounters extreme conditions, either extreme cold or dehydration, the tardigrade is able to enter a state that is called the TUN state, T-U-N, in which the metabolism lowers to less than 0.01% of normal, and the water content of the body drops to 1% of normal. And there's an illustration here on Ask Nature that sort of shows how the tardigrade is producing this sugar solution, which is entering its cells. The water is forced out, and then that sugar essentially encapsulates all the sensitive tissues and DNA in the organism's cells. So tardigrades are really interesting because they are animals. They're not like little bacteria or anything. They are between 0.5 millimeter and 1 millimeter in size when they're full grown. And what's really interesting, what's really cool about that is that you could actually find samples of tardigrades potentially in your school environment. They're very common. They live in lichens and mosses all over the world. So you could go out and collect lichen and moss, rehydrate it, and potentially extract some tardigrades and look for them under relatively low-powered microscopes. You don't need a super high-powered microscope to see them. And we have a video that shows what they look like under magnification, and also shows them going through that process of dehydration and rehydration. Do you want to cue that up, Abby? So this video doesn't have any sound, so I'll just talk over it. So you can see, I think this is time-lapsed, so it shows it shriveling up into that desiccated state. And while the tardigrade is in that ton state, they can withstand really extreme temperatures and conditions, including radiation and vacuum of outer space. They've actually taken these up to the space station and stuck them outside without any protection from the harshness of outer space. And they've successfully rehydrated and even reproduced after returning to Earth. So you can see they sort of puff back up and go back to their business. So if you want to collect some of these to share with your students, we have a link. We'll be sharing some links to some of these resources and pages that we're sharing. But I found some instructions that are relatively simple for doing so. You can use like a simple, like a knife to scrape lichen off trees or stones or other things that you might find in your schoolyard. You can collect moss, whether or not it's dried or fresh, and you basically just soak it in water. You want to use non-chlorinated water if possible, so rainwater, distilled water is best, and basically let all of that material rehydrate, which would include the tardigrades if you've been able to collect any. And then you actually pour out the excess water, leaving your moss and your lichen samples behind, and then you squeeze or shake those over another transparent dish or something that you can get under your microscope. And that will, if there are any tardigrades in your sample, they'll be clinging to and in that moss and that lichen. And so, by discarding the excess water and then sort of wringing out your moss sponge, essentially, you'll be most likely to collect some tardigrades. And then you can look for them under just in like a basic 100x power lens. You should be able to see them, and once you've maybe identified them, could pipette them up onto like a glass slide and get them under a stronger powered microscope if you have one available to look at them more closely. And again, we'll share some links to where you can see sort of a step-by-step of how to do that, and some further images of kind of what you're looking for if you have kind of a field of organic matter in your microscope viewing area, and what the relative scale, what these little guys would look like if you had any in there moving around.
Gretchen, we had a question about how long does it take the tardigrades to rehydrate? Do you have any idea?
I have seen references to 30 minutes to a couple hours. I think the recommendation for, if you're trying to collect your own and want to have the greatest success looking for them under magnification, would be to maybe just let it soak overnight.
Awesome, thanks. And thanks for that question, Andrea. Are we ready to go back to my screen, Gretchen?
Yeah.
Great. We were going to shift into talking about the design challenge just for a minute, but if anyone has any other questions about this lesson, the African midge or the tardigrade or anything, now would be a great time to ask it. Alright, so I'll take that as a not now or not yet, and we can launch into the design challenge. So Gretchen and I have discussed about making a future webinar focused completely on the design challenge as we get closer, but we wanted to make sure that you all know what it is and know how to find those lessons, because those are also new since some of you may have logged into the platform Gretchen, did you want to just give a really high-level overview of what the challenge is?
Yeah. So the materials that we created for this curriculum are super adaptable. You could use them to address any design challenge of your choosing, but we are encouraging folks, if it works with your student population and your education goals, to consider participating in our Biomimicry Global Design Challenge, which is a global, worldwide challenge, as the title would suggest. And this year we are focusing on climate change. And the challenge has two categories of entry. There is a student-only category and a category that's geared more towards professionals and entrepreneurial, largely university students, folks that want to pursue developing their technology for market if successful. But the student challenge is really focused on learning and applying a biomimicry process. And the student category of the challenge is open to both university and high school students. And I want to encourage you not to feel discouraged or dismayed about high school students competing with university students, because this is such a new field that really the vast majority of our participants are learning and applying biomimicry for the first time when they participate. And our high school participants have performed quite well. And in fact, our first place winners last year were a team of young women from a high school in Canada. So that's pretty cool. So, I'm trying to think what else I should say specifically about the global challenge. All of the submission requirements for the global challenge are addressed in the curriculum module that we created. You can choose, of course, to modify the deliverables you want your students to create if you're not going to have them submit to the global competition. But if you do use the curriculum in full, it will help your students produce the necessary submission materials, which include a short video pitch for their idea. Sort of explains what the problem it was that they were hoping to solve, their design idea on how it was inspired by nature. And then they also submit a illustration or photo of a prototype or something else that provides visual representation of their design solution, and a presentation document that most people use Keynote or PowerPoint to put together. It's a 12-page maximum sort of overview of their design process, how they applied biomimicry to solving the challenge that they chose, and what their ultimate solution was, and sort of how it stacks up to some of the key criteria for a biomimetic innovation. So,
the deadline for this is April 30th. That's right, Gretchen?
Thank you for thinking to add that. Yes. So April 30th.
I think if you really want to go through the process thoroughly, it's a great time to start thinking about it. And what's great about the 7 design challenge lessons is you're not just telling your kids to tackle climate change through the lens of biomimicry, which is totally overwhelming to them, but this scaffolds the process for them and really gets them to use kind of the human-centered design process to apply concepts from biomimicry and from nature to tackle this specific challenge. So I encourage you to check out those 7 lessons. Our teachers use them a lot with EcoRise programs to tackle other challenges, and with great success. So they are there and available for you. And I did want to mention that if you know other people, other educators or people in the biomimicry world, they are available for download free of charge from our website. So they come kind of complementary with our science curriculum, but at our ecorise.org/shop, anyone can download them, just the design lessons. So if you know anyone and they're interested in applying, or you have students who want to go rogue and do it on their own, they can go on the website there and just download them themselves.